Yeah.
All right.
So we can just go through it, talk about it.
Yeah, just start and talk, start going.
All right. So an introduction, if anyone's watching this is a project proposal, informal presentation on my summer project with DevoWorm. And I'm going to be looking into simulating embryogenesis, specifically with C. elegans. And DevoWorm is a subcommunity of the OpenWorm community. And I'm here with Bradley Alicia and Tom. Is it Tom?
Portuguese.
Yeah. So it's produced for these communities, and I'm going to go over a little personal introduction for myself to sort of introduce myself to anyone in the OpenWorm community watching, and also give details on the proposed project that I'll be working with. So quick background about myself. There's me. I go to Worcester Polytechnic Institute in Massachusetts. I'm a rising junior. I previously went to Phillips Exeter Academy. That's really where I got my start in computer science for sure. And I'm interested in getting a research career, not necessarily with academia, but definitely in pursuing a PhD or staying around in research fields. There's all sorts of innovative things in between computer science and biology. Academically, I'm majoring in computer science with a double major in bioinformatics and computational biology. WPI offers that. It's a new major. And there's plenty of fun courses in there that they're experimenting with. And I have a couple of my courses listed below that sort of offer some background into where I'm coming from with biology. Just sort of I have experience with the intro level courses, molecular biology, cell biology, and have a feel for the necessary bio background to do a project like this. So really quick background. So this project that I'm proposing, the simulation, it's sort of based around conceptually agent-based modeling to some extent. And I've done another project, which was a simulation on the spread of disease through a population of college students. So it's a little different than a cellular simulation, but conceptually it functions in a similar way in that you're simulating individual atoms of a simulation, individual cells or units. I just have some pictures from that slide just as a little tidbit of background information. So this project, though, simulating embryogenesis in C. elegans, this is what I have sort of come up with the help of Bradley Alicia as a way to get involved in the community and start exploring this project. And the goal of it is going to be to produce a functional simulation of C. elegans embryo development. And we're not sure how rigorous I'll be able to do this. It's sort of an exploratory project in that I really want to navigate the existing tools, CompuCell3D specifically, which I'll talk about in a bit. But I want to sort of test the validity of different ways of simulating this. So why and how are we going to do this? It's a great project to go about right now because it's important to validate existing models of development. And it's also a great way, this sort of simulation, it's a great way of exploring means of discovering new hypotheses. So what these new hypotheses are, couldn't go into specifics. I don't know, I'm a little, Bradley, are there any details you want to fill in on about why and how at this point?
Not really. I think at this point, just to say, this looks pretty good. You're going to get more details as you get into it.
Yeah.
Because, there's certain things we might be able to do with CompuCell3D, and then there were certain things we might not. And I think one of the things this summer would be to find out or to figure out like what things can we do really well in that environment. Because, you could produce a bunch of code and then, other people could say, oh yeah, here's the code. Here's what we can do well in this simulation. And then, we can build on that from there. So it's really just a kind of a survey of this platform.
Yeah. Okay, for sure.
Yeah.
Have you used that before, Bradley?
I'm just a little bit familiar with it, so I'm not sure.
Okay.
I've seen them use it like in different projects.
Yeah.
As far as using it for C. elegans specifically, so it's supposed to be like a way to model cells and tissues. So what you do, you can add in different types of data, you can add in different features, and then they simulate. It's like a physics simulation.
Yeah.
But it's agent based, so you have individual cells interacting. But the question then is like, how do you make it work together? It's not trivial, I mean, you have to really, depending on your organism, depending on your data.
Yeah, and it seemed flexible at different levels. It definitely seems like a flexible platform in that you can extend it with sort of the base like Python scripts within, like you script a simulation with Python, but you can also modify the core modules, all open source, with C++. You can write plugins if you want to get really specific about how you're editing it. So, to go into a little more of the biology background about what's motivating the DevoWorm project. So this right here, this graph, which is on the screen, is a cell lineage tree. And at the very top, you can see in tiny text is zygote. This binary tree shows mostly binary tree shows the differentiation from a single cell to 968, I think it is, cells of C. elegans worm. And this has been around. This graph and this process is pretty well studied of knowing which cells go and become which tissues in this development process. So this is the background of what further models are sort of built on. However, the reality is it's a pretty complex process to go from one cell to a full organism, and like most things in microbiology, it's not understood very well. There's a lot of mysteries and a lot of uncertainty about what's going on. These images right here on screen, these were taken from an online video showing a time lapse of development in C. elegans. So you can see going from one cell to two on the left, going from two cells to four in the middle. The timestamps are on there. And on the right image, you can see sort of the worm structure starting to form. It's bending a little bit. You get the more complicated, fully developed worm starting to form. I thought these images are actually really cool to look at. And the whole video, watching the whole video, it's about 5 minutes. You can see that development happen. It's pretty cool.
So the previous diagram showed the time it was taking for the cells to differentiate also, right?
Yeah, it's an unlabeled graph, but it is based on time.
Okay.
Yeah, so a note here on this part of the upper part of the tree is the embryonic differentiation, and these other parts here down further, these are post-embryonic. So like the worm actually grow, it's an embryo and it differentiates and then it hatches, and then during its larval phase it actually generates cells as well. But that's
of course, right.
Yeah, I mean, so but the idea is that this entire tree is, every adult has depending on whether they're a male or hermaphrodite, they have a certain number of cells. There are a couple differences in the male, but it's always generated the same way from organism to organism. So.
Yep. That's why it's called a mosaic, right?
Well, yeah, some people call it mosaic development because it's that sort of deterministic type of setup.
Yeah. This is where Bradley's expertise comes in handy, and where my background knowledge sort of falls apart. So differentiation trees is another way of representing this growth or differentiation from one cell to an organism. And this graph recognizes the spatial element of division, and it's based around some of the work of Dr. Gordon and his theories of differentiation waves. Though I am certainly lost on the details of this, I have taken a look at some of the papers, but I definitely feel like I need to get that book of Gordon's and read through to get a better understanding of what's going on here, to be honest.
Yeah, we have that on YouTube too, don't we, Bradley?
Which one?
I mean, like you're explaining this. I remember you gave us talks. I don't know, were they recorded? Probably not.
Yeah, well, some of the journal clubs for OpenWorm are recorded, so the YouTube channel should have links to those, or like the OpenWorm YouTube channel has. I can send Josh links. I don't know if you've seen those talks. We give talks to the OpenWorm group every couple. I think we've given like 3. They're just kind of like reports on the project. And so that's where, it's like an hour long, and it's like, okay, this is what we're doing. So that might actually be helpful to like get some ideas.
We record those things.
Yeah, we have it on YouTube.
All right. I've looked at some. Maybe I've heard explanations of it and read explanations of it. But it's certainly something that maybe like seeing one more, like if it's a video that I missed that went in particular depth.
Yeah.
Then yeah.
Well, this stuff is pretty subtle, and we can actually talk about this more over the course of the summer, because we actually built a differentiation tree for C. elegans up to a certain point, like just trying out like using a data set to build one. So we actually have one. It's not perfect, of course, but the basic, we can go over the basic idea later on. But yeah.
It's not. That's sort of the impression I got. Is it's not crucial at earlier levels of this modeling. Like right now, the next actions are really to explore CompuCell. So as the project progresses, this will become more important to really bring a focus back to this. But for right now, I think this is just something to keep in mind in background and something to pay attention to and learn for myself. So yeah. Everything's good here, right? Yeah. I want to make sure. So CompuCell3D is, we've been talking about this a little bit, the tool set we wanted to use for this. And CompuCell3D, from their own description that they have, it's an open source simulation environment for multi-cell, single-cell based modeling of tissues, organs, and organisms. And this image that you see on screen here is an example of one of their cell sorting simulations, which they have built in, or as an example model that you can check out and run. And to just show sort of a little bit about what the interface looks like. And so it uses stochastic processes to simulate cell movement and gradient diffusion. There's an inherent randomness like temperature jitteriness to the cells, which are occurring at all times at every step, and that causes a lot of the cell movement, and that's important to create a realistic simulation. Well, it's not necessarily, it's how it creates that process of cell movement. And the simulation environment itself is pretty flexible. We talked about this before. You can extend it with Python scripts to describe cell behavior and XML configurations to set up a particular simulation the way you want. And it can also be extended by creating your own plugins or editing others' plugins. You good? Yeah.
Yep.
Okay. Sorry for a little Google disconnection. So I think just to sort of summarize this talk, not summarize the talk, but to talk about where I want to move forward with this, I think the next actions for myself are really to, as I said before, get familiar with CompuCell3D. I have a quick start guide, which they have themselves, which covers a few different ways of making simulations. So I'll sort of go through those this week and really see what I can come out knowledge-wise, explore the topic. And I think checking out the plugins, the C++ stuff, user code, check out the source code better, understand how it operates. Is another step to check out once I'm familiar with the basics. And I think as the project evolves, I want to coordinate with Bradley Lish, coordinate with other members to determine specifications for a simulation. Because I think this is part of a longer-term goal to create a more robust simulation, which is doing this. And this project itself is exploratory prerequisite in a sense to be able to move forward sort of broader project.
So do you anticipate that we're going to have to
what was that?
do you anticipate that we're going to have to write a plugin?
I mean, I think that it will be useful to understand how flexible it is to modify the environment.
Right.
Yeah. I'm not sure at this point.
Okay.
Yeah, I mean they have I think they have like a development community, pretty active one. So there's that, and then I don't know. I mean, they have a group of people working on it. I mean, if we have any problems that we don't know too much about, we can always ask them for their opinion. I mean, as far as I'm concerned, this looks pretty good. I mean, I think that's probably a good pace for the summer and for beyond, just to get a sense of like, what can be accomplished, and then doing things that are, extensible that we can build on, that other people can use. And then, really, I think keeping it simple is best. So, don't try to like conquer the world with it. Just kind of like get, something simple going. Even if it isn't a C. elegans thing, if you can get a small animation going, maybe something you think resembles C. elegans, that would be good. A good starting point. Like Tom's stuff on Morphozoic, he was doing some work on just taking like pictures of a C. elegans embryo and trying to find patterns in it. It's just stuff like that. I mean, that isn't like a huge undertaking, but it like gives some idea of what it would be, how it would work.
For sure. I have a question about further visions of this project. What kind of data do you see being used to create this kind of simulation? Like, how would you take real world data? What real world data would you take to once you sort of have a C. elegans simulation?
Well, we have data on size, and we have data on position of cells. We have data, of course, on what the cells are. So, like in the lineage tree, every one of those cells is identified with a specific name. And then you have, after that, you have other data that's attached to that. So we have size data, locational data, and then each cell itself has data in the literature on. Remember, we were talking about that the first meeting you attended. There's data on gene expression. There's data on just the general behavior of the cell. There's a lot of information we can incorporate. And I'm not quite sure how, well, we'd have to try different things. So that's going to be a challenge too, is how does the data integrate into the model? Like, if we have a data set of cell location, can we build cells, individually that then, divide and then end up in the right place, things like that.
Yeah. Okay.
But like I said, it's probably best just to get familiar with the model first. Start small and then work your way up. And then we can start trying to integrate data and see where we go.
Yeah, I wonder if it uses something like in graphics, like you have these keyframes idea, where if you want to create an animation, you create a bunch of frames that are spaced apart in time, and then the software will interpolate,
Oh, yeah.
I'm just wondering, maybe they have that. I don't know.
I don't know. I think from my impression, it's like the cells are the units of analysis, and you can simulate them over time. So maybe, you have like an initial condition and you have some parameters that are, I don't know exactly.
Yeah, I mean, if you say like at time 0, these cells are here. Time 1, they've moved here. Time 2, or time n, they've moved here, and then sort of it'll come up with some way of moving the cells around so they get where they're supposed to be.
Yeah.
I don't know. So, I mean, but that doesn't violate the physics of it.
Well, yeah, because you'd just be integrating.
Yeah. Something like that.
Yeah.
I don't know. Just wondering.
I don't know. I don't know how they achieve that technically, but like.
Well, they use it in animation all the time, but so.
Right. Especially in a system like this where it's there's this deterministic component. So most development isn't really deterministic. It's sort of stochastic, like in the human embryo. A lot of the cells will respond to signals. So if you have cells that divide and then they respond to a signal locally and then they differentiate. And, there's some degree of what they might become, but, it's really dependent on the presence of different factors. So they can do these experiments where they, take a little piece of tissue, in an embryo, put it somewhere else, and it'll change its fate. Things like that. But that doesn't happen in C. elegans. The C. elegans embryo is very deterministic. It's, if you can actually take an embryo, at like the 2-cell stage, split it apart, put them in separate places, and then those parts of the embryo will differentiate as like 2 separate halves of the embryo.
Really?
Yeah. So it's quite different than what we're used to in humans, and I think for what they've used this for. But I think I sent you a list of readings, and one of the readings was where they simulated a chick embryo, and so that simulation, maybe instructive, is a lot different than what we're doing in C. elegans.
Yeah.